In this video we will discuss about car evolution. Though in one of our previous video we have discussed about different generation of car, we will discuss here in this video with some more details compared to the discussions that we have done previously about different generation of cars. So before directly going into the different generation of car, let us understand how these T cells get activated naturally. So the one of the ways by which T cell is activated is this TCR signaling or the T cell receptor signaling, and the major signaling factors is this item signal, and this item signals will leads to the activations of the intracellular signaling pathways such as NFAT, RAS, MAPK, JNK, P38, MAPK, and this is occurred because of this dimerizations of the zeta, and these things will cause the activations of the T cell receptors. And there are other ways also by which these T cells get activated. One of them is co-stimulatory domain activations, such as, and this will trigger the activation of P13K, which is the survival pathway, and the TREF NF kappa beta. And the third signaling pathway that cause T cell activations naturally is the cytokine signaling. That is, cytokines come and interact with the receptors. That leads to the phosphorylations in the intercellular part, and that will trigger the activation of JAK1, STAT3, 5 pathways. This is one of the example. So the researchers or scientists, what they did is they try to mimic these signaling pathways by making a synthetic construct, and the first time they are able to develop this is with only using this CD3 by replicating this TCR signal ITM signalings using CD3 zeta that can be seen here. So the first generations is only with the CD3 zeta, and however later on, to increase the anti-tumor efficiencies of this CAR construct, they have introduced cytokine signalings, replicate cytokine signalings, and also the co-stimulatory signalings. So this is in general how the CAR signaling looks like. In the extracellular domains, they have this binding domain and the linker for the flexibility of this single chain fragment variable, and in the transmembrane domain they have this transmembrane region CD28 or CD8 alpha, and thereafter they have the co-stimulatory domain. It could be one or two. Commonly used co-stimulatory domain are CD28 41 beta 41BB, and then they have this cytokine signaling inducer, that is NFAT, that will enhance the anti-tumor efficiencies of this activated CAR T cells, and the CD3 zeta is for the activation of the T cells. So now we will go through the different generations of the T cells. So it might be a quick revisions for the people who have went through the previous videos in which I have discussed about different generation of CAR. So in the first generation, since I have told that they have only tried it with this activation replicating item signaling pathways by using the activation intercellular domains as the activation of the T cells, it is CD3 zeta. So once this single chain fragment variable interact with the antigens that are present in the cellular domains or on the tumor cells, it will trigger the activation of the T cells. And thereafter, to enhance the efficiencies of these CAR T cells, they have also introduced this co-stimulatory domain. So once the extracellular domains interacts with its corresponding antigens, it will not only activate T cells, but it will activate T cells not only through the CD3 zeta but also through this co-stimulatory domain. And in third generation, they use two co-stimulatory domain, and in the fourth generation, along with the co-stimulatory domains and this activation of the T cells using CD3 zeta, they have also used this NFAT, this nuclear factor of activated T cells. It is the immune modulators or the cytokines that are released, and that will also trigger the or helps in the anti-tumor efficiencies. And the latest is the fifth generations, where they have used inducer such as IL2R, truncated IL2R. So what it will does is this inducer will trigger generations of the cytokines such as IL2R, which will enhance the activations of these T cells and the immune response against the tumors or the cells, cancer cells with the antigens which are corresponding to the binding domain that are expressed on the CARs. So these are the different generations. In the previous in the one of in the early video where I have discussed the CARs, I have not discussed our fifth generations. So in this video, this is one more additions. The fifth generations are also been tested, in which the inducer is used between the co-stimulatory domain and the activation T cells domain, that is CD3 zeta. So these are different generation of CAR and how they mimic, or how they got the idea to mimic the activation of the T cells from the nature, and what are the signaling pathways that they are trying to, the CAR Ts has replicate, and also in trying to replicate in future using the fifth generations. That's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.